I completed a PhD in biochemistry in 1971. The Navy promised me that they would put me to work in a biochemistry laboratory, and it turned out to be a clinical chemistry laboratory run in support of a toxicology program, the United States Navy Toxicology Unit, that was in Bethesda, Maryland. I subsequently worked in that position for 8 years, and then left to join the Air Force as a civilian employee at the Armstrong Aerospace Medical Research Laboratory in Dayton, Ohio. I worked there until 1989, and then I came to a position here at what was called the Chemical Industry Institute of Toxicology. After that, I worked as a consultant and then as a faculty member at Colorado State University in the Department of Environmental and Radiological Health Sciences. At Colorado State, I was a professor. I taught toxicology and had a research program. In 2002, I returned here to North Carolina at the Hamner Institutes to be the division director in the Division of Computational Biology. And today I have that position, and I'm also the director of our Institute of Chemical Safety Sciences. From my background, it's clear that my primary experience is with toxicology testing as it was done in the 20th century, the first 30 years of my career. Those tools were developed back in the 1930s. They've been refined over time, but they've been refined much as a patchwork quilt, adding tests, but not taking a careful look at whether there were ways in which we could develop improved testing methodologies based on new technologies and a new understanding of biology. There are several key technologies that were emphasized in the toxicity testing report for understanding toxicity pathways. Toxicity pathways is a description of the key signaling pathways in cells that can be disturbed or perturbed by chemical exposures. One of the key requirements is to have ways we can test for those perturbations in toxicity pathways using cells and test tubes, in vitro testing with cells. The three technologies then are in vitro biology, in vitro test methods, computational biology based on an understanding of toxicity pathway function in these cells, and then in vitro to in vivo extrapolation. So toxicity testing in the 21st century, to some extent, is a terminology that comes from that report. The report committee consisted of 21 individuals, and I was one of the members of that committee. So toxicity testing in the 21st century looked at new technologies, a new understanding of biology, and asked the question of how toxicity testing should be done in the 21st century, and how new tools should be used in the interpretation of toxicity testing results to make estimates of safe regions of exposure for people. But after we see these responses in vitro, we have to have a way to interpret them. What will happen at lower doses? What will happen at concentrations that are found in the environment? And how do we go from understanding what happens to a cell at a given concentration to what will happen in the environment where people are exposed by amounts in food or in air? We've created a proposal for accelerating implementation of the recommendations of the National Academy of Sciences report. The intention over the next year or two is to raise money for a program that we would use a series of examples, prototype examples. We take chemicals that have been well studied, where we have knowledge of their pathways of function, their toxicity pathways, and have some previous information on their toxicology from animal studies. We would take the chemicals, study them in vitro, develop the computational tools and the in vitro in vivo extrapolation tools to do a formal safety assessment. That process would look at assay design and a series of 10 pathway prototypes over the next 5 years, showing how the process would work, and in our minds accelerating the path forward by providing a clear roadmap for how information could be collected and could be used.